Welcome to the first Pint of Science podcast. I am Matthew Davidson. I'm the Science Communication Director for Pint of Science. With me today are Jonas Verhellen, who is an applied mathematician at the University of Oslo, Vegard Vinje, who works at Simula, and Professor Leif Øyeheim at Oslo Met. Today's topic is applied math. And so this is something that I think a lot of people are not super familiar with. I mean, everyone goes through mathematics, through grade school, but usually, I think most people and most of our listeners have probably stopped well before the point where you all are. And so I'm hoping that through this discussion and through our conversations today, people can get a really nice sense of what it is you do and how applied math can apply to a lot of different topics. So by way of introduction, I'd like each of you to just tell me why you are excited about your work. And maybe, Leif, we can start with you.
Okay, so first of all, I'm just an associate professor.
Associate. That's right.
So I do a lot of teaching. So I really enjoy that. I have students who are, to a varying degree interested in mathematics, but reaching out to them I find very fascinating and interesting. And then I have I have bachelor students mostly, and also master students who are a bit more interested in the subject. And supervising them is really my joy, and supervising them in their projects and so on. So, but in my research, my topic is mathematical modeling in biological systems. So for the most part, I work with modeling calcium dynamics in heart cells, which is very relevant or very important. These calcium ions in heart cells are very important for actually maintaining a sound heart, so that everything goes as planned in these cells. And although, of course, I like doing things that are relevant for an applied field like biology or medicine, but I also like the mathematics behind it, and also making numerical simulations on the computer and see what happens when I sort of tune the different parts of my mathematical model, and when that is actually relevant to what the people doing the experiments and the medical doctors are interested in, then that is when I really enjoy my work.
Thanks. And we'll obviously get more into all of these things. But by just to introduce yourself, Jonas, what makes you excited about the work you do?
Yeah. Well, thanks for the invite first. What makes me excited about the work I do is the impact I can have. So originally, I'm a theoretical physicist. That's where my math background comes from, and hence I'm very interested in using mathematics, like I think the three of us, and just for the beauty and the joy of doing it. And in the work I do now, I focus on also like biophysical models, but instead of heart cells, I look at brain cells, neurons, and I did specifically for major mental disorders, so schizophrenia and bipolar disorder. And then I do some work in artificial intelligence for drug design as well. And in my modeling for neuroscience, what makes me really excited is how close we can get to what actual patients are going through. So these are complicated diseases nobody really understands. And we can try to take genetics and try to model that into actual things we can measure in a human brain. And that excites me.
Mm-hmm. Cool. And then very good. Same question. What is exciting about what you do?
So I think for all the three of us, I think sort of our worldview is the same, in that we feel that we can understand the world if we can describe it in equations. So this is, I don't think other people than mathematicians think in this way. So if we have a system like, I'm working on modeling of the human brain. So if we can describe what's going on in and around the human brain with mathematical equations, then this is our way of understand what's going on in the human brain. And if we can understand what is going on there, we're much more likely to find drugs or treatments that would actually help patients. And this is of course what inspires me in this work, that this very or relatively theoretical approach that I think we all have can actually help people that have no clue at all what's going on behind these models. So that excites me a lot.
Cool. So the three of you all mentioned models, and this is something that comes up not just in mathematics, but all across science. We have like climate models and we have disease models. So, and I'm not sure everyone really understands what that means. So could you maybe just explain, like, what do you mean when you say I'm modeling the brain or I'm modeling a disease?
Yeah. So what a very simple example of a model would be Newton's law of motion, right? So that when you push an object with a force, you can predict the acceleration of that object. So in essence, if you know the forces acting on an object, you can determine what the movement of that object will be. And this is a very simple model of how reality works. So it's a kind of an approximation of what you will actually see. Of course, you make some simplifications when you assume that you can accurately compute this movement. But it's a very good approximation of reality. And when you start adding on different factors of other forces acting on this object, then you will increase the complexity of your model. So I think starting with an equation to describe reality, that is a model.
Yeah, I think you can think about it in a way like a blueprint an architect has. It is not the house, it's a drawing of the house that has a lot of information about the house. And the more complicated your drawing, your technical drawing gets, the more you know about the actual real object. That's what we do, but then in a much more mathematical language, which can describe much more than just a simple drawing can do. It's that simplification and then adding of details that was mentioned before. That's really key.
So I was thinking of the models that have been used during the pandemic, because before making the model, you need to make some assumptions, which probably are simplifying the whole thing. So in order to make something that is not too complex, so making the models, they are called SIR models, that are used to describe how infection spreads in a population. One way of doing it is actually to use methods from chemistry. So you actually assume you have a population or people in a population that mix sort of ideally, like chemicals in a petri dish would. And they will react, and that reaction is then sort of the analogy to infection happening. So one actually assumes that the population is kind of like, or divides themselves into more kinds of molecules, which you can then describe by equations. And then you get a very simple model formalism, which is based on simplifying assumptions. And that's a very common way of making models. And I guess we always do that. But it's different depending on what you model. But there are always many simplifying assumptions to begin with.
So is the first step kind of trying to figure out, like looking at a situation, trying to think of like, what is this like? What does this look like? What equation, like what system is this similar to that I can base it off of?
What are the key components, I think, is the main question. And once you identify that, you can indeed look at existing systems from different fields that are very relevant, or you can start from scratch and build up your own systems using the mathematical background you have. And the more mathematical background you have, the more systems you could describe, or the more complex your descriptions could become. Although that's not always necessarily better. You have more leeway.
Yeah, I completely agree. And I guess it's like you use the tool that is available, that you know how to use. So if you don't have a hammer, you use a shoe, if that's what you have. So it's not necessarily the perfect tool, but if it works, that's okay. And I guess also that's this quite famous quote by, I think, statistician George Cox, I think his name was. All models are wrong, but some are useful.
Okay.
And which I think maybe is a bit extremely put, because I would say maybe all models have their limitations. Some are more accurate than others, but most of them can be used to predict something or say something about the system.
So what does it look like to go from, a simple model like you were saying, like Newtonian physics, to something like trying to model like a mental disorder? Like what is involved? I mean, that seems like a huge step. So what is involved in building a model from like Newtonian principles to a mental disorder?
Yes, I would say that you always build upon work that's been done before. And in many cases, I think the first step is the hardest. Coming up with Newton's law was fairly impressive, I would say. And then what we do in neuroscience, and to some extent also in cardiac cells, is you make a model that describes an aspect of the cell. In neuroscience, it's typically how the electricity that goes through the brain, so the brain sends out signals in terms of electricity, how that propagates to the brain. You make a model of that, and you just start off with something very simple. You know how a wire conducts electricity. Okay, you start off with that, and then you start seeing what are kind of the batteries and the pumps that influence the wire, and how can you make a model that kind of gives you the same level of electrical behavior that a neuron does, a brain cell does. Again, this is not an accurate model of what a neuron is, but it gives you one aspect of what it does. Then you start with this, you compare this to healthy people, to healthy neurons, and then you start looking at, okay, what if I start changing things here? How do I break the system? And can I figure out if the things that break the system come from the genetics that we see in people with severe mental disorders? That's in my case, but I'm sure there's similar levels of increasing complexity.
Actually, many of the equations that are used in modeling of the human brain as a mechanical object is based on just Newton's law of motion. But the difference is that Newton's law of motion is typically defined with the point forces. So if you put force on an object, like with just a point, then that object will move. But obviously in the brain, the forces are different everywhere around the brain. So essentially, you have to solve this point force problem over a million different small points around the brain. So this is essentially what you do, that you solve Newton's law of motion in 1 million different places around the brain.
Okay.
So it will give you 1 million equations with 1 million unknown. And obviously, we all need computers to solve these equations, right?
So what did you look at exactly? You're talking about mechanical properties of the brain?
Yes, so we are modeling fluid flow around the brain and also going through the brain.
Yeah.
So fluid flow around the brain is just fluid flow in open spaces, while fluid flow inside the brain acts more like a sponge or a porous medium. But Newton's law of motion is the basis for both these types of flow.
And is it then for like what happens during sleeping? Or
yes. So it's also changing during sleep, this fluid flow. And we have modeled this a little bit, and there's experimental data on that coming out right now or last year. That this clearance of waste products is a little bit faster during sleep than the awake state.
What I find, which is common, at least the two of you, is that your models are based on quite fundamental physical laws. And that is also quite general, I think. And all of us, actually, we work in biology or with models applied to biology. And even there, laws of physics are often the basis of the models that we use.
That's something I really enjoy about the sciences, is that, I felt like in college it was very much split out, like you're a biologist, you're a chemist, you're a physicist. But then, at this level, you're all working together, and like the math is applied to the biology, and you're using physics. And I always find that's very exciting. What is it like working with, you're all mathematicians. What is it like working with biologists and physicists and other disciplines?
Well, I have some, actually, for almost decades, I've been working with biologists, and especially when I worked on my PhD. There was a lot of collaboration with biologists, and it's not straightforward. It of course depends on the biologist, but often they do not. Obviously, they do not have the same background in maths as I did, But then there are those quite important people who have some background in maths in addition to in biology, and they are very important because they are sort of the mediators between us who are mathematicians and the biologists who often are. I guess it's a bit like you said, Vegard, about us and understanding these systems through the equations, whereas the biologists obviously do not do that because they have sort of the proper background in the subject and didn't learn it through equations like we did. But I guess I find it a very nice way of learning about a system through the models and the equations.
Do you think that's a trend in science? Are we going to a more collaborative, interdisciplinary system?
Yeah, I think it is happening. For instance, in my work, I briefly mentioned also do some research in AI for drug design, and there the opposite thing happens. We actually take ideas from biology and put them into mathematics. And so we have algorithms based on ideas of evolution, but also like that you have different species in different places, and this can teach us a lot as well. And I think this interdisciplinary approach, I share an office with experimentalists. It is very, very useful, and not always in very straightforward ways. You have the big projects where you work together and talk together, but you also have the small things of being around people all day with a different background, and there's all the small things you learn about the practicalities of what somebody does day to day. That teaches you a lot, I think.
I will add that when I am down at the place I work at Simula, I cannot call myself a mathematician because to them I'm not a mathematician. I'm a computational modeling guy.
Okay.
So I don't do all the theoretical analysis and these things, but more into computational modeling. But obviously, when I go to our collaborators at Rikshospitalet, then I'm a mathematician, and they think that I'm a mathematician. So maybe this is a bit of an identity crisis.
Right.
It's very recognizable.
Yeah.
Absolutely agree. Yeah.
But the collaboration with the hospital at Rikshospitalet that we have, it's very fruitful. But I think it took about 10 years before we spoke the same language. And from the collaboration was initiated to papers started rolling out and actual results were produced. So it takes some time before the language is the same.
Yeah, sure.
Yeah, I'd like to add on that. We have a collaboration with Ullevål Hospital, also but mostly Ullevål Hospital, and it's the same. It's very interesting. You come in a completely different world, especially once it gets really medical. But there is a lot of work into translating, and that takes hours and time. And it's a whole different language all over.
I guess they have always used statistical methods and statistical models, so they're very used to that. But that is a very different discipline, really. Although it uses a lot of mathematics, of course, but that's something they use to analyze their data. Whereas we begin in the other end of the problem. We use what is known about the system and make the equations based on them. So we approach the problem from the opposite end. As the experimentalists who do the experiments and then analyze the data. So there's a very important distinction between the statistical approach and our modeling approach.
Yeah, it's a very good point. It's indeed, I think we've all experienced.
Great. So I want to ask you a little bit about how you got into this. Like if someone is listening to this and they're starting to think like, wow, that sounds really cool, I want to get involved. Like, what is your story for how you got involved with this, and what would someone who's listening to this and excited about it, how would they get into this field?
I got into it through a lecturer at the University of Oslo, Hans Petter Langtangen, that sadly passed in 2016, I think. He was a brilliant lecturer, and he had one lecture where he actually showed us what he did in his workplace, and it was regarding blood flow. And modeling of blood flow in the body, and that was really inspiring. So the year after, I applied for a summer internship at Simula, where he worked. So that was my way into it. And for people that wants to do this, I think there are several groups at universities everywhere that works on biomedical modeling these days. So it should be possible to approach this subject through any educational institution nowadays, I think, in Norway. I'm also teaching a bit at the high school level, and I also always try to tell them that the stuff that they learn there is actually relevant for what we do. These are things that we do every day, like taking derivatives and use the logarithmic functions, and it's used so much that you cannot even believe it when you are in high school. But it's hard to see at that point in time, I think.
Yeah, very interesting. I think what was mentioned before with the vacuum you live in high school where everything is separated. And it is indeed surprising feeling. The more research I do, the more I look back at the basics and use those more and more. My story is a bit different, a bit more complicated, but I'm originally a theoretical physicist, did a bachelor's and master's in that, but I was always kind of interested in the computational aspects as well. So my theory work was really pen and paper and more or less nothing else. But on the side, I like to do some programming. And then I got into fluid dynamics, and I did that for a while, then did an internship in the UK in that during a summer just for fun. And that computational aspect always kind of stayed with me. And I did some actual research in physics for a while, and I decided to change careers. And I moved to London, worked in the industry for a while, and I was in a pharmaceutical company where I was doing deep learning, so AI for drug design. And then you get surrounded by all kinds of people with very medical or biological backgrounds. And that's kind of how I got back into the academic world, and now in neuroscience, which is relatively close, I think, to physics with all the first principle approaches using fluid dynamics, electrodynamics, all these things you can build up from relatively simple principles. That was my path.
Yeah, so my way into it was I wanted to study mathematics, and I didn't really make up my mind regarding whether it should be applied or not until I got close to doing my master's, and then I guess a bit coincidentally it became an applied master, oil related. This is way back in the 90s. So I guess I would have made a lot more money if I went into the oil industry after completing my master's degree. But instead, a bit coincidentally as well, I think I got a note in my shelf at the institute about not a vacant actually, but the possibility for a PhD position at the Agricultural University of Norway at Ås, south of Oslo. Later on, I've been in different jobs, but for the most part, I've been modeling, as I mentioned, these heart cell dynamics, but also actually brain cells, neurons, and so on. I'm doing that as well. And it's really been very interesting and fascinating.
So what are the medical conditions that you're investigating?
So not really what I did for the not really medical conditions. I guess it's more about studying or trying to make sense of experimental data using mathematical models. And what actually I did, and it's not become too technical, but I did something in the brain or brain-related things, where I was part of a team that studied these water channels in astrocytes.
And how calcium affects?
Not calcium in that case. That was more of these. So that very interesting with the Ole Ottosen's team at the University of Oslo. So very, really collaborating with these very, very clever guys.
Relatively close to the work you do, actually.
Yes. So these channels that you mentioned, they are the bridge between the water outside the brain and the water inside the brain.
Yeah.
So water flow through this channel is extremely important for exchange of substances in and out from the brain. So these channels probably increase flow when you sleep and help clear out the brain of waste products. So
we are now beginning to talk collaborations here.
That's what I was hoping we would get to, actually. Because one of the things, you all talk about collaborating with different people in different fields, but the three of you are all in similar fields, but doing different, applying to different subjects, or at least somewhat different subjects. And so I'm excited to see, now that you've kind of talked about a little bit of what each of you do, what sort of overlap do the three of you have with each other, and what sort of collaboration do you think you could do?
I think at least we have some people at our lab that are working on modeling of both fluid flow in and out of brain cells, and also including these bridge channels between the inside and the outside of the brain. So that would, of course, be very relevant for what you are doing.
Yeah.
But me, myself, I mainly rely on what they tell me about these channels and results. I don't sit down too much side by side by them and develop these kind of models. But I do collaborate with them, definitely, and their work definitely overlaps with what you have done.
Yeah, I think that's a key point here. That, I mean, there's 3 of us here, but I'm sure we all have, if we start talking longer, we all have colleagues that know each other and work together, and it's much more integrated. We're somewhere on the spectrum of people in and around Oslo who do neuroscience related, applied mathematics.
So starting collaborations is, as you know, right, it's quite difficult. At our research lab, it's always this goal from higher above to have people collaborate more within the research unit. But it's remarkable how difficult it is to just sit down together and do research if it's not a complete overlap. Something needs to be founded before you do that. And
it's an organic thing most of the time, right? If you find collaborators sometimes in places you wouldn't expect.
I guess it also has to do with personal chemistry and all these things. So I of course work in an institute at the university where we mostly do teaching. We discuss a lot between us, but not very many of us actually collaborate on research. So because we have different backgrounds and we discuss whatever we find interesting, but not many papers come out of that, unfortunately.
Not directly, I guess. It's all this informal talks that are so important, and I think that's very hard to describe to somebody who's not actively, professionally doing research. Is that talking to the person sitting next to you in the cafeteria can be extremely important for what you're developing without it directly influencing a paper. And it sculpts how you think about things. Sometimes you learn about a small technique here and there. And these are the things that are really important. Why conferences, for instance, are also important. I mean, they're still going on online now. But the important thing of a conference is typically not the talk, but the talk after the talk.
Yeah, so then the online conferences has been a disappointment. In my opinion, but it's a bit of personal preference, I think. I'm looking forward to going again.
Remember somebody once saying that if you see a couple of people standing around a table and writing something on a napkin, it could be the lunch order for the next day, or it could be a Nobel Prize winning paper. And that's kind of the key thing about actually doing research for a living. So it's a very human thing.
It's a little bit different in our group, actually. That I think we are about, let's say in our research group, we would be maybe 10 researchers or something like that. And I think everyone has at least 2 or 3 people that they work closely with in that group.
That's quite nice.
So that's quite nice. But this is within the research group, but there are obviously different groups that it's hard to start conversations.
But in an educational institution like where I work, most of us working there we have our own research which we did before, and then we bring that with us, and everybody did that. So there's no normally there's no sort of policy when hiring people that they should have the same background as those people already there. So that doesn't. So then the research at the institute wouldn't then necessarily benefit from a new person coming in. Although it could, but it depends on the people meeting each other and finding common interests. And they do, of course, it could be papers coming out of it, but normally not, unfortunately.
So as far as the sort of papers coming out, we talked about kind of how you got into this and what it is you do day to day. I want to give you a chance to kind of talk about like what is the application, the applied part of the applied math that you do. in your main studies, what have you found? What are some cool results or applications of your work?
Yeah, so what I'm working on, it was, I think we might have talked a little bit about it, but about 10 years ago, they discovered that this fluid flows inside the brain differs between sleeping and awake mice. And they found evidence that the fluid actually flows through the brain and washes out waste products that accumulates over the day when the brain is active. And many of these waste products are also the same waste products that are associated with Alzheimer's disease and Parkinson's disease.
Mm-hmm.
So it's very associated, but it's not clear that it's like causing these diseases. But then that caught our interest in the research group, and I think we started trying to model these systems in 2015 or something like that. And the field is relatively new, so if you can model something about it, then it will give answer questions that were not known at all before, because the field is so new. So, but then again, things move very rapidly as well at the same time. So in our research, we have actually, in collaboration with Rikshospitalet, we have performed and quantified these experiments in human beings for the first time. So it means that we showed that the results that they found in mice, that fluid washes through the brain, it holds also in humans.
Okay. So is this another reason why we should be getting enough sleep at night?
Yeah, so I think there's lots of reasons for why we should get sleep, right? But and I think sleep is obviously good for you, but we are trying to look more into the mechanisms of what's going on during sleep. So sleep is obviously good, and our study doesn't say that sleep is good or bad. It's just trying to figure out the mechanisms behind the sleep. And in addition, it's quite a debate on what is driving this fluid flow through the brain. So originally, it was postulated that it is the heartbeat and the cardiac pulsatility that pushes the fluid through the brain. Because when they hold the vessels, so they don't pulsate that hard, they don't see that much tracers entering the brain. But through computational modeling, we showed that also breathing affect this flow. So this fluid flow will pulsate both with the heart and with the breathing in a relatively comparable manner.
So is the one of the goals, I guess, then to understand the mechanisms of diseases like Alzheimer's?
Yes, the mechanisms of fluid mechanics in and around the brain, which can be linked to both sleep and Alzheimer's and other diseases. Now, recently, we are working on a project. We're trying to link this to cancer as well. So there's obviously some fluid disturbances in tumors within the brain, and we're looking at possibilities to reach these tumors with drugs through this fluid system around the brain.
It's very interesting.
So normally the drugs will be tried to be distributed through the blood.
But then you have the blood-brain barrier.
Yeah, and you have a blood-brain barrier, which limits the drug distribution to the brain. But in theory, it should be a little bit distorted around tumors. But in practice, a very little amount of drugs actually enter the brain through the blood. So we're actually looking at data from injections of tracers in the fluid around the brain and see how it distributes around brain tumors. From the preliminary data that we have, it seems like it's actually accumulating a little bit around tumors. So this is very interesting to
hopeful.
Yeah, it's really cool that you can actually you can use your model and then actually see some applications, some results from it. Yeah.
So we have to make a little bit of a distinction here, because the data that we get, where they actually inject tracers, that shows that fluid or particles will accumulate a little bit around the tumor region. But based on this distribution of particles, we can sort of reverse engineer to try to find the fluid fields that governs this movement.
Cool.
So you cannot actually measure the fluid movement,
Mm-hmm.
but you can use your model to get the fluid movement.
Got it.
Mhm.
Cool. And so you were talking about similar brain modeling, right? But
Yeah.
you're working in
Mental disorders.
mental disorders of the brain. So
Yeah.
is that a similar kind of modeling, or what have you been doing?
Yeah,
with that?
we focus much more on the electrical signals.
Mhm.
So the difference between the housekeeping of the brain, where you have to make sure that enough things go into the brain for it to work and enough clearance of waste products happens, is something slightly different, also using basic physical principles and what we do, where we look at what's the information processing that happens in the brain. So people who have bipolar disorder or schizophrenia, their brains tend to process things differently. And this could be from hallucinations to mood disorders, but it's really about how does the brain work on an information processing basis. Now, what we do is we have models of neurons of brain cells that can conduct electricity in healthy settings, so for healthy people. And then we try to use more or less every source of information we can get about schizophrenia and bipolar disorder to make changes to these models. And I work for this interdisciplinary project called Forment at the university. And what we do there is we look at a cohort of patients, of Norwegian patients, and we really try to get as much information about the specific patients as we can. So there is patient histories, what have their lives been, what kind of medications have they got, which diseases do they have specifically, or which symptoms? It's a big question in mental disorders. Is it a disease or it's a combination of specific symptoms? That's unclear. And then brain scans. And then what's really, really interesting is on one hand, they take blood samples of them to try and get an idea of has anything changed like on a biochemical level in their brain. Now, the problem with blood samples is that if you eat a lot, or it's a certain time of the month or whatever, the levels of certain products in your blood can change drastically. So it's not really a fixed or good measure for what is happening in the brain. And so what they're doing at Ullevål Hospital now is they've taken a select group of patients and controls, so healthy people, and they've taken skin biopsies, so a little bit of skin, and then you add chemicals to it, and you can bring that back to stem cells, so to the most primary form of your cell. And then if you give it the right kind of nutrients and chemicals, you can start growing this back into brain cells. So what they're doing there is they're growing brain organoids, which are literally tiny brains or tiny brain-like structures in a petri dish.
You're making brains in a dish.
Okay. I have colleagues who they won't be happy if I call it brains in a dish, but yeah, they're brains in a dish.
But that's what I say. So your computer models are informing these brains.
It's the other way around. So
okay.
so that they grow these brains from healthy people and people who have certain mental disorders, and then they can see what's the difference. And the nice thing is we can look at it on a genomic level. So they do experiments on them to figure out what the genomic changes are between the two groups. And then we can make a model of how that should change the electrical properties of the brain. So the brain fires, even those tiny lab-grown brain things fire electrical signals. And we can make a model of, okay, we think this genetic change is going to affect our model in this way. So the combined electrical properties of an entire brain, which you could measure with these brain scans on living people, which we also do, should be different in this way. But now we have direct access to a version of the brain. And then you can start
that's really cool. So
putting electrodes
what have
into
you
it.
been able to find from that?
So well, it is ongoing. So growing this,
of course.
Yeah, the problem is growing these brains is almost like having a baby. It is 9 to 11 months of growing them, and it is extraordinarily difficult and expensive. It's a very expensive baby.
My understanding, I don't have any, but my understanding is that babies are very expensive.
I can assure you brain organoids are as well. But also very, very impressive. I mean, these are very, very new things. Just keeping them alive is hard. And one of the reasons is getting fluids in and out, because they don't have veins, for instance. We're not at the point where these brains have veins. So you have to keep them small because you have to keep nutrients to them. So the whole question about the fluids comes back to that. And then, the genomic part is done. So we're aware of what the genomic changes are. We're looking into how to model them. And now we're waiting for the measurements of how the electrical properties change. That's very intensive. I mean, it takes several people to do this by hand.
Yeah, and I imagine the computers, well, I guess for all three of you, the computers you use are not, it's not like laptops, right? You're using pretty expensive
You
computers.
can do quite a lot with a laptop, but of course, I guess
Okay.
in some, I mean, to test your code and so on, you use the laptop. I don't know how about you. Maybe you have
Most of-
scale simulations.
I use the laptop to log into clusters.
Yeah. Okay. So you
But
use
most of
clusters.
the work is on the laptop,
Yeah.
and then if there is, if you know that this is the right simulation that
Exactly.
you want to
Yes.
run, then you do it on the cluster.
Yeah.
So, but you need to do lots of testing on a computer or a laptop before you run it on a supercomputer cluster.
If it's possible.
Yeah.
In our case, we're very bound to large scale simulations,
Okay.
which.
And like you're talking about a lot of the history of mathematics and modeling, yes, and the way to teach it, yes.
Yeah. Well, I don't actually work with that a lot, but I do teach it a little bit, and of course I mentioned these SAR models for epidemics.
But you're modeling. You're talking about the different species and stripes and.
Yeah. I mentioned that, and I didn't. Let's be honest, I didn't really make a very big contribution to that field, although I found it very interesting to work with, and it did give me a PhD. But of course, you sort of circle around the main topic because you're not really able to do something that someone the other ones already contributed in the field didn't.
What are you talking about? Every PhD thesis is groundbreaking.
No. I guess it is at the moment you had it in, but 25 years later, it's no, not that. 20 years later, it's not that great. But I did get the PhD. So what if I was to mention one sort of contribution, and the one that I'm most satisfied with, I guess that would even be in brain science as well, because I mentioned these water channels that I worked with. As a follow-up study of that, I made this model which combined the neurons that you mentioned, but also the astrocytes. It's sort of the other brain cell. What I found was all of a sudden I guess I had this aha moment where something happened that I didn't expect. So because I found out there were the ion concentrations. So you have potassium, you have sodium, and chloride, and they were starting to oscillate, and I didn't expect that in my model. And so working sort of looking more closely at that phenomenon, I realized, of course, someone else had seen that before, but of course my model was the first. Nobody had used that model before, so I could say something about how these astrocytes, these sort of the other brain cell, affects the dynamics of ion concentrations around the neuron, which might be at least related to epilepsy, and have something to do with epilepsy and the dynamics of epilepsy. So it's not like cited a lot, but there have been some. So yes, I was pretty happy with that.
Yeah,
but these
make our little contributions.
These ion concentrations, this is what drives the electrical signals through the brain, right?
Sure,
absolutely. Yeah, so there's a whole interplay where if your fluids around the neuron don't function properly or are disturbed in some kind of
way. So what there's this positive feedback. So if you have a lot of activity around the neuron, there will be a lot of potassium, which again drives the more potassium out, and then you have this situation of going into something that is epileptic. Yeah.
Well, I think one of the cool things I'm gathering from this is, we started talking about this Newtonian model, and I'm thinking about, like, with physics, you start with like a Newton model, and then you can like add in friction, and you can add in air resistance, and you can keep building and building. You can add in like deformation, and you can add in relativity, and eventually you get up to this like universe model. It sounds like, with the three of you were talking about, you say, well, you start with just a neuron, and then you have these connections, and you have also the mechanics and the electrical and the fluid and the ion channels. So I can kind of see how you could start with this very basic equation and then build up to epilepsy or bipolar disorder, something like that. Is that the right kind of way to think about it?
Definitely. I think if you put all the three of our models together, then you would be much closer to a real brain model.
Yeah, it's starting
And
to get there.
it's actually quite could be integrated, and I think we are working a little bit on trying to make that happen. But you sort of have to make the pieces for the full model to some extent correct before you try to put them together.
And of course, the computational costs become much
Sure.
larger, right?
Definitely.
So how far are we from having like a brain in a computer?
It's super far.
Quite
Still pretty far.
Although what does exist is you've got a place called the Allen Institute in the United States, and what they've done is they've taken I think it's a mouse brain, could be a rat brain, I always mix the two up, and they have a small part of it that processes visual inputs, and they have a fairly accurate model of that. But just running a small, it's a really small part of it, that takes a supercomputer, just to run a few seconds of the activity in that. And there's no astrocytes in there, there's no fluid dynamics in there, it's purely a version of the electrodynamics of it. And that takes a supercomputer, quite a large one to run. So that's the scale you should be thinking of. Do that times 3 or something, and then do that for the entire brain, and then you've done a mouse, maybe.
Okay.
So, but I think whole heart models, they do exist, but it's also like that. If you have a mouse heart, it's of course a lot less computationally intensive to solve. But the heart is relatively well understood compared to the brain.
Yeah, it is.
The brain is terribly complex.
But you still have to stick to continuum models.
Yeah, because you have billions of cells that you cannot represent all of them. I'm not sure about mice, the mouse hearts.
I think they have tried that actually to model. But this is like sort of the research front that you maybe are able to resolve all the cells in the mouse heart.
Well, I think we're going to start wrapping it up. It's been a little bit. I really appreciate the three of you coming and talking to us. The last question I'm going to have is I just want to hear what is one of the coolest things you've encountered, and think about that for a second, because we're going to talk about Pint of Science, which is happening all over Norway. It's going to be the 9th to 11th of May this year, in bars and pubs and cafes around. We have Oslo, Stavanger, Bergen, Trondheim, Tromsø. So we have all over Norway. I think some of them are going to be streamed, so even if you can't make it to the in-person event, you can see it. Again, that's the 9th to 11th. We'll have more information on our website and how you can register. We're going to have a whole bunch of different topics. So it's going to be and a bunch of different topics and speakers in each location. So just as the last question, real quick, each of you can go through what is the most exciting things you found and you've done in your research?
For me, it was definitely the moment when we got back the results from the sleep versus sleep-deprived individuals in the human study of that. I actually myself quantified and analyzed the data, but I didn't perform the statistical analysis of it. So when I looked through all the data, there's just lots of different images showing where these tracers are localized in the subjects, and I couldn't tell like a really big difference between the groups just by flipping through all the data. But then sending it through the statistical analysis, we didn't expect there to be any big differences. But when it came back, it actually turned out that we were actually able to reproduce the results from mice 10 years ago. So that was a really aha moment for me, or really, really cool moment.
Yeah, for me it was also data related. When we got back the genetic analysis of these brain organoids, these brains in a jar, we saw that the genetic changes that we see in the two groups were genetic changes known to be important in the early stages of the disease, really, really early stage of developmental. So these brains are actually young, and that I found very cool because they do grow over 9 months. And then you look at them, and the genetic changes are more or less the ones you see very, very young in patients. So these brains are actually young, and that's I found to be very cool.
Yeah. So I already mentioned one thing that I thought was pretty cool, but I also maybe the first sort of discovery I made when I worked with these pigment patterns was looking at a model of production of melanin in mammals. Using my model, I found that animals tend to either produce the black melanin or the yellow melanin. So when you look at a tiger, there's a very distinct pattern of black stripes on a yellow background. It could be the opposite, of course. But so I still consider that to be my sort of coolest moment, because it does alienate very well with what we actually see in these animals.
Cool. Well, thank you again so much for spending the time. I'm going to put up your contact information, if that's all right. If people are interested in following up with you, if they want to ask some more questions, if they'd like to maybe get involved in this kind of research. So definitely check that out. Check out Pine of Science, May 9th through 11th, all across Norway and around the rest of the world. We have videos from last year and everything as well. So yeah, thank you guys so much. And hopefully we'll do another one of these again soon.
Thanks
Thank
for having
you.
us.
Thanks for having me.